Hello, hello. Today we're going to be talking about genetics and inheritance again, except for this time we will be focusing on mutations and sex-linked traits. So some key terms. A chromosome is just a tightly coiled strand of DNA within the cell nucleus, but there are actually two types of chromosomes. So there are autosomes, and these are going to be your 22 pairs of chromosomes aside from your X and Y. Now, because of this, you can actually have two different types of traits. You can have autosomal traits. These are going to be controlled by genes on one of the 22 pairs of human autosomes. You can also have sex-linked traits, and these are going to be traits where it is controlled by a gene located on a sex chromosome, so your X or your Y chromosome. And sex-linked traits have recessive alleles on the X chromosome. Because males only have one X chromosome, if they have the recessive allele, they will express the trait. Women are afforded a little bit of extra protection. Of course, this is not always the case, but because we have two X chromosomes, typically if you have that recessive allele on one of your X chromosomes, it will be masked by the dominant allele on the other X chromosome. And these are going to be things like male pattern baldness. So when people say that you should look at your mother's dad rather than your father's dad to kind of determine when you might start to go bald, it is because it is a sex-linked trait. Now, there are other forms of baldness, so this is not always true. But for natural occurring baldness, this is typically the case. It's also going to be related to like color blindness. So if you didn't know, men are more likely to be colorblind than women are, and it is because it is often a sex-linked trait. Now, we've talked about natural selection as a mechanism or a force of evolution, and we've talked a little bit about mutations. We're going to go into more detail on them today. So a mutation is a change in the genetic code that creates entirely new genetic material. This is really important because it is the only mechanism of evolution that actually creates a new source of alleles. So it is the only thing creating new variation. Now, we talked about how mutations can be negative, positive, or neutral. Now we're going to talk a little bit more about how they are actually caused or created. So they can be spontaneous mutations, and these are going to be mutations that result from natural DNA copying mistakes made during cell division. And these can't really be predicted or prevented. However, there are induced mutations, and these can be protected against and predicted because they are going to be things that are caused by exposure to ionizing radiation. So this is where when people talk about, being careful and taking precautions if you have to get a lot of X-rays or if you are out in the sun a lot. Specifically with the sun, when you get a sunburn, it actually damages those cells, and this can end up resulting in an induced mutation that results in cancer. They can also be caused by exposure to chemicals called mutagens. So the biggest thing that people refer to with this is going to be like your carcinogenic chemicals. So when people talk about not smoking because it is a carcinogenic drug, it is because it can actually lead to cancer, which is a mutation of a cell. They can also be caused by infections through viruses. Now here at the top, you can see a point mutation where thymine is being replaced by cytosine. And as a result of this change, valine is going to be produced instead of isoleucine, which actually changes the protein that it's going to code for. Now, cytosine, guanine, adenine, and thymine are base pairs, so they follow a specific pattern and they attach to each other in a specific way. Sometimes these point mutations actually result in the production of the very same amino acid, so nothing is actually negatively being replaced, and the same protein is going to be coded for. These really have no impact on the organism's phenotype, and these are called synonymous point mutations. However, in the example that we see here, where the amino acid is being replaced by a different amino acid, this is a non-synonymous point mutation, and this change in the amino acid sequence has a very dramatic effect on the actual protein being produced and what it does for the organism. Now, there is another type of mutation, and this is going to be the most common for induced mutations. This is shown at the bottom, and it's called a frame shift mutation. In this case, a nucleotide is actually being inserted into the gene, which shifts all of the other nucleotides down by one. Now, nucleotides are actually read in groups of 3 called codons. So shifting them all down by one actually dramatically changes the DNA sequence and how it's read, which results in an entirely different amino acid and therefore entirely different protein being coded for. Genes can actually copy themselves and be inserted into different regions of the genome. When this happens, they're called transposable elements. But what's really important to know out of all of this is that a synonymous point mutation is a lot less dramatic and dangerous than a non-synonymous point mutation. And a non-synonymous point mutation is less dramatic and dangerous than a frame shift mutation. Now, another form of mutation actually takes the form of chromosomal duplications. And aside from like cancer-causing induced mutations, these are probably going to be what most people are more familiar with. One version of this is trisomy. So this is a genetic disorder where a person has 3 copies of a chromosome instead of 2. These are going to be on your autosomes, not on your sex chromosomes. And the most common example that people are actually going to have some introduction to is actually trisomy 21. This is where you have 3 copies of your 21st chromosome, and it is actually what causes Down syndrome. But you can also have duplications on your sexed chromosomes. For example, Klinefelter syndrome, which is a genetic condition where a male is born with an extra copy of the X chromosome. Now, intersex people, there are a lot of versions of sex chromosome duplications. So you can have XXY like Klinefelter's, you could have XYX, you could have XXX. There are a lot of different versions. Klinefelter's is a little bit more common, and we know a little bit more about it. Now, we talked last week about positive, negative, and neutral mutations. Trisomy and Klinefelter's other duplications are considered negative mutations. Now, this means that they are negative if you were looking at it in a broad view. If you're looking at humans just as animals and like evolution-wise, them needing to reproduce in the most efficient and productive way, these are negative mutations because these individuals oftentimes are not going to be as fertile. They're not going to have an easier time just living life. They might have other health issues, sexual preference issues, and as a result, they are not going to be having offspring or as many offspring as a quote-unquote normal individual without these mutations would have. However, that does not mean that they are inherently bad. People with trisomy 21, people with Klinefelter syndrome, and a lot of other duplication issues can go on, especially in today's world, to live very happy and relatively healthy lives. So it is not that they should be looked down upon or negatively in social connotations. It is just that their life is harder in some ways because of these mutations. Therefore, they are considered negative mutations. Now you guys should watch this YouTube video. There's also a couple activities for you to do this week. You do not have to screenshot your results and submit them with your lab assignment. Do the color blindness test. Don't do it. It doesn't really matter to me because it's not graded. However, I do recommend that you do the chromosome activity. It's pretty interesting. You actually get to kind of experience what it's like to be a genetic scientist and pair up chromosomes. And whenever you get done with each case, it will actually give you a review of it that explains the individual and the limitations that their life has as a result of the genetic disorder. You can also ignore all these due dates. I have not updated them from last semester. Now, because this is a fairly short lecture, we are going to go ahead and move on to the Lab 4 lecture on Hardy-Weinberg. Now, admittedly, I am not a huge fan of math, but I have to do this every semester, as well as just within anthropology sometimes, so I've gotten fairly used to it at least. And the Hardy-Weinberg equilibrium is actually an equation that has been developed to help kind of estimate population genetics. Starting off with some key terms, a population is the number of inhabitants in an area, and it is continuously being modified by increases. So these are going to be births or people migrating in, or losses. These are going to be deaths or people migrating out. Now we've talked about genotypic ratios. Now we're going to talk about gene frequencies. So these are the ratio of a particular allele to the total number of all other alleles of the same gene in a given population. Genotypic frequency, however, is the number of individuals with a specific genotype divided by the total population. Now we've talked about mutation and natural selection as forces or mechanisms for evolution. And now we're going to talk about the last two. So one of those is genetic drift, and this is going to be any changes in allele frequencies that occur randomly due to factors like differential reproduction. So this is where things like sexual and mating preferences are going to come in. There are two special types of genetic drift. So one is the founder effect, and this occurs when a subset of a larger population ends up founding the next generation due to population loss or population movement. We also have bottlenecks, and this is when a founding population doesn't actually have the original population's gene frequency, meaning that some genetic information was lost. Now, these can be due to things like part of the population moving away, and the part that moves away has more of the dominant allele, and therefore the remaining population has more of the recessive allele, and that will continue to be passed on at a higher percentage than what the original population would see. With the bottleneck, this is often going to take the form of like a disease spreading through that part of the population doesn't have a natural immunity to through their genes. And so then they are going to be the surviving group whenever they go on to create the next couple generations. You're not going to see the phenotypes that were killed off because there is nothing to keep pushing them forward in the evolutionary cycle because they've all died off. So that information has been lost. We also have gene flow, and this is the exchange of genes between previously isolated populations that then begin to interbreed. This is going to be things like if you think about breeding different dogs. Let's say you have like a lab and like a golden retriever. they've been separated for a while, enough that, you know, they have two completely separate populations. They don't interbreed, but they are still part of the same species. Well, let's say something like human intervention or encroaching on the territory or an increase in predators or a decrease in food pushes these two populations closer together than they were. And then we start to see them interbreed. Now we're going to start seeing a new admixture of both of these breeds' alleles into a new breed. Now, the Hardy-Weinberg equilibrium is used to predict the probable genotype frequencies of a Mendelian trait in the next generation. But there are a lot of conditions for something to be in equilibrium, and it never actually occurs in real life. So the conditions are that there is no mutation occurring, there is no natural selection occurring, there is also no gene flow occurring. So the mechanisms of evolution are not in place. There is also not, the population is also going to be infinitely large. So we're going to see an equal number of deaths and births each year, and there is not going to be any kind of migration in or out of the population. The mating in the population is going to be completely random. All members of the population are going to mate, and all members of the population are going to produce the same number of offspring. Now, obviously, this is never true. You cannot keep mutation, natural selection, or gene flow from occurring. You also can't keep a population infinitely large and stable. Regardless of what kind of organism it is, there is always some sort of sexual preference or mating preference. So that's kind of out the window. And organisms never have the exact same number of offspring within each individual in a population. So these are never true. You might be asking why it's even important that we have this equilibrium or this equation. But it is actually because it helps us predict populations and population changes, as well as kind of keep tabs on populations that might be in need of a little bit of help or preservation. Now, if all of these conditions are true, then the genotype frequencies for the trait should be the same from one generation to the next. We aren't going to see any change. But since that isn't true, we have a lot of variation. And this equation is actually used sometimes by like farmers and other breeders of animals. It's also actually used by anthropologists to determine and kind of predict past human ancestor populations. So obviously, there is really no way for us to know what the Neanderthal population as a whole looked like. But we can make some assumptions using this equation. Now, there are actually two equations within the Hardy-Weinberg equilibrium. So there is p squared plus 2pq plus q squared equals 1. And there is also p plus q equals 1. Now, p is the frequency of allele A, so the dominant allele. Q is the frequency of allele A, or the recessive allele. P squared is the frequency of the homozygous dominant individual. Q squared is the frequency of the homozygous recessive individual. And 2pq is the frequency of the heterozygous individual. So here in this example, we're going to imagine a plant population where flower color is a Mendelian trait with two dominant alleles for white flowers and red flowers. So the flowers on these plants may be white, they might be red, or they might be speckled. And we know in this population that we have 75% of the alleles are going to be white and 25% are going to be red. So P equals 0.75, Q equals 0.25, because we want to convert those into decimals. And then we're just going to plug them in to the P squared plus 2PQ plus Q squared equals 1. Now, sometimes you might only be given the dominant allele, so you might only know P, but you can still determine the rest of the equation because we know that P plus Q equals 1. So we're just going to take 1 minus P, and we're going to know that Q is, in this case, 0.25. There are other instances where you are only going to be given the genotypic frequencies. So you might be given 2PQ or Q squared or P squared. In that instance, all you really have to do is take the number that you're being assigned for, and let's say you're given P squared. And if we use this example here, your P squared is going to be 0.5625 or 56.25%. To find P, all you have to do is square root that 0.5625, and you will end up with your P value. From there, you can figure out your Q value and just start to plug and play. So after we've done all of our math for this example, we can see that our genotype frequencies on the second generation are going to be 56.25% for the white, 37.5% for our heterozygous, and 6.25% for our red. If we see these frequencies in the next generation, the trait is in equilibrium. But if we don't see these frequencies in the next generation, then the trait is evolving and it is not in equilibrium. There is a video here you should watch. It will actually go over the Hardy-Weinberg equilibrium. It may be better detail than I can. Of course, ignore these due dates. There is also another video that will teach you guys how to do those sex-linked Punnett squares, which you do need to know for the lab. But of course, if you have any questions on this or the lab 3 questions, shoot me an email and we will go over those. I am going to remind you guys, though, that the Hardy-Weinberg lab 4 questions are a participation lab. So as long as you guys answer them, include the work that you've done on them, so the steps that you've taken, you will get full points for it, even if your final answer is not correct. Mostly because I think it's just a jerk move to expect you guys to get all of those correct when you likely have not done this equation ever before. But also because I don't really want to go through step by step and grade each step to make sure you guys are understanding that. But of course, if you guys have any questions, you can let me know. We will go over that. This is a really good lab for the discussion post. You guys can put it, put the problem in the discussion post and maybe work together on some of those to help figure those math problems out. That would be totally acceptable in my opinion. And I can give you guys some tips and answers if you do. But that is all I have for you guys today. So I will see you next week with some more lectures.